Good afternoon, everybody, to what is a very beautiful afternoon and going to be filled with an exhilaration of information. My name is Payal Mukherjee. I'm an ear, nose, throat surgeon, a co-founder of Beyond Science. This webinar series is part of University of Wollongong's fortnightly conversation to bring together researchers, industry experts and thought leaders every fortnight. Today, we are going to be discussing how research and collaboration is tackling global challenges, specifically in the area of vision restoring surgery. We're going to hear from some exceptional colleagues as they discuss innovation and corneal bioengineering. This also presents a significant manufacturing opportunity for Australia, and we're excited to discuss the progress in knowledge exchange and translation to make real-world impact. Before we start, we want to acknowledge country, and we're just going to watch a video.
We acknowledge that country for Aboriginal peoples is an interconnected set of ancient and sophisticated relationships.
The University of Wollongong spreads across many interrelated Aboriginal countries that are bound by their sacred landscape, an intimate relationship with that landscape since creation.
From Sydney to the Southern Highlands to the South Coast. From freshwater to bitter water to salt. From city to urban to rural.
The University of Wollongong acknowledges the custodianship of the Aboriginal peoples of this place and space that has kept alive the relationships between all living things.
The university acknowledges the devastating impact of colonisation on our campus's footprint.
And commit ourselves to truth-telling, healing and education.
So today I'd like to introduce you to my exceptional panel. Firstly, we have Professor Gerard Sutton from the University of Sydney. Gerard is a highly experienced, innovative and celebrated ophthalmic surgeon. Professor Gordon Wallace, who's well known to the university, he is he's an Australian Laureate Fellow and my partner in crime in the Beyond Science program. Last but not least, we have Danielle Fisher. She is an experienced public health professional and a long-standing general manager of the New South Wales Organ and Tissue Donation Service. So thank you everybody for being here. We encourage members of the audience to submit their own questions using the Q&A function. We'll try to get through as many questions as possible, but first we're going to go through the talks. We're just looking forward to listening to Gerard first. So Gerard, take it away.
Thanks, Payal. G'day everyone. It's a great pleasure to be part of the UOW Luminaries webinar series today, and we'll be talking about Bienco. And I think Bienco, most importantly, is a story of the importance of research collaboration and where it can lead. We're talking about the impact of Bienco on global blindness. And blindness is obviously a major issue for the individual, but it also causes social and economic significant ramifications around the world, to the point where the WHO and a number of organisations have made that a priority in terms of trying to address it so that we can have an impact around the world. The particular blindness problem that we're focused on is corneal blindness. The cornea is the clear window at the front of the eye, and when it gets injured or infected, it causes scarring, such as in the picture here. And at the end stage, the only real treatment available to us is corneal transplantation. But the problem is that there's a global shortage of donor corneas. And so our proposed solution is to meet this unmet clinical need with a bioengineered cornea. And there's no doubt that regenerative medicine and cell therapy are really becoming very important in trying to find problems, solutions, I should say, to the medical problems. Right now we have over 23 million people around the world who are corneal blind, but only 1 in 70 will actually receive a corneal transplant. And there are over 12 million people on waiting lists around the world. Our solution is to build this cornea which will replicate the human cornea to a point where we can have unlimited transplantations available. I'll talk a little bit about Bienco, the organisation. The name comes from a combination of bioengineered cornea, and it's made up of a consortium of world-class organisations within Australia: University of Sydney, University of Wollongong obviously, Organ and Tissue Donation Service, Queensland University of Technology, the University of Melbourne, and the Centre for Eye Research Australia. And our vision is to eliminate corneal blindness globally, and we intend to harness not just the best Australian technology, but all the expertise that is included in this consortium. And we want to be the first to market commercially with a bioengineered cornea that will have an impact on corneal blindness around the world. Just a little bit of background. The cornea is that clear window, but it's comprised of 3 major layers. So it's a good target for bioengineering. It doesn't have any blood vessels. It has 2 layers of cells on the inner and the inside and the outside, and a stromal layer in the middle. There are different transplants that we use to treat this disease. We can do a full thickness transplant, or we can do a partial thickness transplant. And Bienco will be producing products that will address both these needs. Bienco is part of corneal transplant history. It was 1905 when the first human corneal transplant was performed, and Australia was, pretty quick off the mark, having the first successful transplant in 1940. And our consortium members have played a significant role in the history in terms of establishing biobanks, University of Wollongong in terms of creating the corneal stroma. And so we have a good history together, and as a consortium we feel that we can bring that expertise to fruition. We have experienced leadership. I acknowledge Mark Daniel from the University of Melbourne and CIRA, and of course Gordon Wallace, Greg Chow and Damien Harkin, our scientists, and Danielle Fisher leading up our commercial and regulatory team. I'll now just point out that we are also globally connected. We have medical advisory experts from around the world in this area, as well as commercial communication and relationships already established. Obviously, these things are important if we want to actually get our product to the people who need it. As someone once told me, a successful experiment in a laboratory will not cure blindness. We need to commercialise in order to be able to get that product to the people that need it. I'll now hand over to Gordon.
Thanks, Jared. So Jared has already explained or described to you the cornea. It's an exquisite structure with 3 different cell types distributed in a 3-dimensional entity, which gives an amazing combination of properties. Those properties being optical transparency, mechanical robustness, and of course harnessing and being able to accommodate all of the biological function that the cornea needs to accommodate. If you look at the stroma, that bit in the middle of the cornea, we uncover an even further level of complexity. Within the stroma, there's multiple layers of collagen-based membranes. Now, those collagen membranes are actually made up of aligned collagen fibrils, and then they are organised orthogonally in alternate layers within the stroma. So a very exquisite structure that's developed over obviously many, many, many generations. Can we bioengineer the cornea? Can we in some way replicate what biology has achieved with those alternate layers accommodating those stromal cells? So let me focus on the stroma for a moment and on the innovative fabrication methods that have become available to us in recent years. This method assembles collagen molecules in an electric field. So we have collagen molecules in a solution, we apply an electric field, and at that isoelectric point in the diagram on the left, those molecules will actually assemble. They will aggregate into collagen fibrils and form a standalone collagen membrane. That fabrication method could be applied in other ways to produce supported collagen membranes and even hollow collagen tubes for different applications. But let's focus on the standalone collagen membrane that we produced using electrocompaction. If we look at that and we drill down using an electron microscope and look at those images, we uncover that we have in fact generated those amazing nanodimensional collagen fibrils that make up those exquisite membranes or those layers of membranes within the cornea. So this fabrication method allows us to replicate the structures that we find in a human cornea. That replication means that we get the mechanical robustness that we need, much more handleable structures, we get the optical transparency, and it's able to support the biology that we need to support. And that biological support is obvious when we introduce the corneal stroma cells into those alternate layers between the collagen membranes. And we look at the viability of those cells, that they're able to be supported over time, and also look at the directionality of those cells. Those cells are growing along the collagen fibrils. And that directionality and the orthogonal arrangement of those alternate layers to get that directionality, that's the combination that gives us the mechanical, optical, and biological properties that we need in the stroma. So that was an exciting development for us and for the Bianco team, for the collaboration that we've been working on for a number of years now. But we can't stop there. We need to take the stroma and we need to build our other layers of cells on top of that. As Jared explained, we need an epithelium layer and we need an endothelium layer. We need to be able to source the cells, and Danielle will speak more about this later. We need to be able to source the cells from a cornea tissue that's donated. We need to be able to isolate and process those cells and to multiply those cells. And we build on a wealth of information from our collaborators groups over many years, our collaborators at QUT and also at University of Melbourne and Sierra. And we have all of this knowledge on how to treat and process these cells so that we're set up to make a complete bioengineered cornea. And that's what we've done. That's what we've done. We've been able to take the stroma that I just described to you. We've been able to take other collagen-based structures and to grow the endothelium layer and the epithelium layer, and then to combine, to build the cornea, hand assemble it on the bench so that it has all of the arrangements of the particular cells that we need and still retain the optical and mechanical properties that we need in that combined structure. So this is a really exciting moment for us, the fact that we could demonstrate we can build a fully bioengineered cornea on the bench. Let me just diverge for a moment. There are other projects that are ongoing in our research laboratories which may also impact on the Bianco project. One of those is the ability to load microcapsules containing particular drugs into the collagen membrane. We've demonstrated that we can use this fabrication approach to get excellent dispersions of those microcapsules throughout the collagen membrane, and then that we can use the distribution of those microcapsules to control the rate of delivery of the drug, the extent of delivery of the drug, and even spatially distribute the drug throughout that three-dimensional structure. So these are parallel technologies that may well feed into the Bianco program as we move forward. But let me move back to the big challenge. It's incorporating all of these technologies into a fully manufactured bioengineered cornea. Now, that's not simple. Right? I've already talked about getting the donated tissue and all of the processes around the cells and the three different types of cells that we need to multiply. I've mentioned the collagen. We will need a source of collagen, a reliable source of collagen. All of the processing options for collagen need to be scalable. And then we need to take those components and integrate them not on the bench and not by hand assembly, by using automated fabrication methods that will generate the bioengineered cornea. And here's another challenge for us. That challenge involves the ability to characterize what we're making. This is unconventional manufacturing. It will require unconventional inline monitoring. We need to be able to look at what we have containing living cells and not destroy it, but just verify that this is the structure we actually meant to build. So there is another parallel area of opportunity and challenge, and we're looking at developing these contactless means of characterization. One is based on ultrasound. We're all familiar with ultrasound technologies in the clinic, but we've been able to take that ultrasound information and build new algorithms that extract that information and interpret it so that we can actually look at the distribution of living cells, look at the proliferation of living cells, and even the activity of those cells in building their own structure within that 3D entity, and all in a contactless manner, and all possibly that can be done in line on a scalable manufacturing process. Let's have another look at that manufacturing process. There's lots of steps here. There's lots of parameters. There's lots of variables to get the fully optimized bioengineered cornea. If we were to take a conventional approach in optimizing all of those steps, it would take a long time, longer than we've got. And so we appreciate this challenge in all areas of biofabrication. Let me take a pretty simple one, and that's just where we use 3D printing to build a biofabricated structure containing living cells. The variables there are similar. We've got our cell source, we've got our materials or our bioinks, and then we've got all of our fabrication parameters. In this very simple example, the conventional approach would require investigation of thousands of printer setting permutations, thousands of experiments. Working closely with Professor Sveta Venkatash and her colleagues at Deakin University, we've used machine learning and customized algorithms to build an interactive process which reduces that to less than 50 experiments. Obviously, this is a powerful approach that we need to adapt in building the manufacturing process for the bioengineered cornea. So where are we now? We've been able to demonstrate we can hand assemble that very complex, that exquisite, that elegant structure that biology has evolved, the human cornea, and we can do that on the bench, retaining all of the functionality that we need, mechanical, optical, and biological. We understand the challenges in manufacturing. We will adapt all of the advances in technology to make our job easier. So big challenges in manufacturing, but we have all of the information, all of the technology to be confident that we can meet those challenges and generate the bioengineered cornea. And I'm going to hand over to my colleague, Danielle, who will tell you about other aspects of that manufacturing process and what's engaged and what other things that we need to engage in to be a success.
Thanks, Gordon. Now I'm going to take you back to the beginning and really start to unpack some of, I would say, in comparison to Gordon, the more simple aspects of the work that we're doing, but I guess also some of the more complex in terms of how we bring this collaboration together. So the first thing that I want to say is, the New South Wales Organ and Tissue Donation Service, in terms of tissue banking, we're never doing today what we were doing 2 years ago, and I don't expect that we're going to be doing the same thing today in 2 years' time that we do today. Our work is around human tissue, and we identify and process human tissue for transplant. Ocular tissue for corneas, musculoskeletal tissue, skin that's used in burns, amnion that's used for a treatment. And we, our role in this collaboration is really the provision of the tissue in the first instance, but also the work that goes along processing that tissue, producing allografts, and allocating those allografts to surgical needs, and hopefully in the not too distant future doing the cell culture work and the production of the bioengineered materials and assembling the layers of the cornea. So here I just show you a quick snapshot of both the tissue bank of the future, the clean rooms that we're going to need to be manufacturing and automating the putting together of the bioengineered cornea, and previously the cells, the grafts that we manufacture today. The not so simple, but in many ways much simpler than the manufacturing of the cornea, is the manufacturing, the Code of Good Manufacturing Practice, and the regulatory considerations that we need to take into account. for the processing of the cornea. So many of you will know that the good manufacturing practice describes a set of principles and procedures that, when followed, ensures that the therapeutic goods are of high quality. And we need to demonstrate good manufacturing process in the biological space for the production of every single graft that we produce. There are immediate priorities that we need to consider and have in place around good manufacturing. We have that at the moment with the simple different corneas that we provide for surgical procedures, and as well as the other grafts. Each manufacturer needs to have an established quality management system. We need to have an electronic quality management system, and we really go into quite an extensive amount of information about our materials. Where do they come from? Are they registered under the Therapeutic Goods Act? Are they registered therapeutic goods themselves? Where do our supplies come from? We need to have arrangements in place, formal agreements in place with our suppliers. We need to audit them. We need to make sure that every single component of our end product is monitored under our Code of Good Manufacturing Practice and also our biologicals dossier. The same for the manufacturing environment. Every process, every step along the way, and every piece of equipment needs to be validated and verified on an ongoing basis. We need to ensure that quality is the foundation of our manufacturing process, and it's not a bolt-on at the end of the process where we have a product. So all along from the beginning to the end, and this is in our collaboration, the role that we play with product release in mind. What we need to consider are in the most simplest things: the packaging, the labelling, the storage, transport and validation. So just by way of example, the labels themselves need to be validated. We need to be able to verify that the print of the label is not going to come off in transport. It's not going to be affected by the way in which the temperature is controlled when we transport an allograft, perhaps in dry ice or in some other way, or even just sitting in a freezer in the lab waiting to be distributed. The packaging needs to be verified and validated on a continual basis, from the first layer that sits around the graft to the final packaging, the box that the graft sits in. Storage, environmental monitoring and controls is really key, as is that monitoring throughout the transport process. And just when you thought that was already detailed enough and we don't want to hear any further about quality on an ongoing and continual basis, we need to be constantly managing for risks, training personnel, validating the training that personnel have had. Each small change in the process requires processes, deviations, change controls, hazard alerts, and ongoing product quality review. On to something a little less dry. For the bioengineered cornea, as Gerard said earlier, there is a global need for corneal tissue. There are 12.7 million people globally waiting for a corneal transplant. There is a fair amount of work internationally in terms of looking at developing a bioengineered cornea. The donor graft is the current gold standard, but as we've identified, only 1 in 70 people who need a transplant, which is the only treatment, can access that transplant at the moment due to the scarcity of the donor cornea supply and tissue banking internationally. There are xenografts available, but they're not preferred by the market. There are a number of other competitors who are working on bioengineered cornea. However, they're only looking at one layer of the cornea. And the work that we're doing as the Bienco Consortium is the only work that's being done that looks at multi-layered, full-thickness bioengineered cornea. So how have we got this far? And Gerard started to talk about, to begin with, the power of collaboration and working together. And for us, this has really been the key to our success. And we started at the beginning talking about the need to play nicely. We've all got different priorities and different agendas and different complexities in the work that we do, but it's really important that we're coming together and working through the challenges as a team. Alongside the teams in the Bienco collaboration are those other stakeholders who we also need to work closely with. So other clinicians and research facilities, patients, investors, governments, etc., etc. Now, just to talk briefly about the regulatory strategy. So the New South Wales Organ and Tissue Donation Service is quite experienced in relation to regulatory products. We have over 250 products currently listed on the Australian Register of Therapeutic Goods, and this is one of the ways that we're going to be able to assist the team to move the products through from the lab through the bench space and into hopefully to the bedside. What we intend to do, we know the regulatory pathway well in Australia. We want to navigate that regulatory process, then move into Europe and Asia, and finally move towards FDA clearance for the States. We also need to consider what our reimbursement strategy will be as we make a bioengineered cornea. We need to ensure that it's going to be accessible and affordable, and we are very familiar with the established reimbursement pathway for biologics in Australia. So from the beginning, we need to be working in that space. Internationally, will require further investigation and to develop collaborations to better understand those markets. So, in terms of value proposition, what does the bioengineered cornea offer? Better patient outcomes, a new sector job creation, industry and infrastructure, health economics and social benefits, and an investment in research which really adds to the sustainability of this work into the future. I guess as I started in the beginning of my talk, what we're doing today is never what we're doing in 2 years' time. And while we're really excited about the bioengineered cornea, and we do expect to have it in clinical trials in 5 to 6 years' time, no doubt in 10 and 20 years' time we'll be moving on to something else. So that investment in research and the generation of funds to create sustainability in that cycle is really important. So in summary, we've really focused very closely as a consortium on corneal disease, the fourth leading cause of blindness, and the lack of access to treatment with the scarce supply of donor corneas. We've identified that there are 12.7 million people waiting for a corneal transplant, that the solution is the world's first multi-layered bioengineered cornea, that we have a competitive advantage because we have the world's finest, or Australia's finest, we think the world's finest, eye research centres together with the finest university research groups as well, that we will be able to use funds and we will be able to access funds for product development, manufacturing, and develop a world-class manufacturing facility where we can continue to learn and develop new approaches. And finally, an exit target where we're going to be able to have products that are licensed for global distribution and trade in 5 to 10 years' time. Thank you.
Wow, so I'm sure everyone attending can see why I'm having a total fangirl moment. Like, this is amazing what you have achieved. Not only has it required a tremendous amount of collaboration, but also funding and also preparing the system for this force to come. We've got some questions coming through. I might just start off with comments from each of you. Gerard, you're sitting in a operating theatre going, my gosh, we can do it better. Most clinicians will stop at that, complain, move on. How do you go about then, comment on collaboration? How do you go about building a team, knowing what skills you need and when in the project pipeline?
Yeah, I think that's a fair question. I guess I do see myself more as an inquisitive surgeon rather than a genuine academic like Gordon, for example. But, surgeons, I think good surgeons are inquisitive. And I realised that there was this need, having done significant work in Myanmar and Cambodia, and that there's just no way that countries, unlike Australia, they just can't access the tissue. And so I think that what I was fortunate enough was to be already in a relationship, a collaboration with the Organ and Tissue Donation Service as the medical director, and I knew that Danielle was running a show that was already successfully getting tissue out and already had expertise in regulatory experience, and also I'd been working already with Gordon on a related but different product, and so it was actually quite easy because the three of us get on well. And, the team that we bring, my senior scientist, Jing Yu, and all the other people from the OTDS and University of Wollongong, we've all been working together. And so that was actually quite easy. But I'm a genuine believer in synergy and serendipity. And I think that this collaboration is an example of both of those. And then we were able to bring in our colleagues from the other institution. That just multiplied the expertise and made the synergy greater. So I think in some ways it was fortuitous, but I think that the first step for me was being inquisitive and just saying, you know what, not only is there a problem in corneal supply, but we have problems such as rejection with corneal transplants. So maybe if we're going to create our own cornea, we can do better. We can actually program it or set it so that it will be less likely to reject, and to be able to produce a product, dare I say, that's better than the natural one. So that were my thoughts, and I think that Danielle alluded to it. It is about a genuine relationship where there's trust, and if there's no trust, then and I've seen it happen many times before, that without that level of trust and building up a bank of goodwill, I don't think that these collaborations can succeed.
Actually, just on that note, just before we move on, one of our participants who asked questions, he's going to be leaving the webinar early, and it's a question for you, and it's something that you touched on. The question is about the expectations and challenges around higher transplant rejection with the bioengineered product.
Yeah, well, I mean, we would think that would not be the case because the cornea is a sort of, in some ways, immunoprivileged anyway, because it doesn't have any blood vessels to recognise, have the white blood cells recognise the antigen. But there's a specific antigen-presenting cell within the cornea, which is the main trigger for rejection. And so we could potentially be printing our cornea with the various collagen layers, with the keratocytes, with the epithelium, with the endothelium, but without the antigen-presenting cell. And we can also incorporate into our model various growth factors that might be able to stop blood vessels growing in, which is a major risk factor for rejection. So there are ways that we can theoretically produce a product which is less rejection prone. So I hope that answers the question.
Yeah, wonderful. Thank you. Next question to Gordon. When someone like me looks at this whole translational research, I'm sort of thinking about how much funding support would be required. Just wondering if I can get from you in comments of the challenges and the successes that you've had, and opportunities in Australia to go from the very early start to develop an idea, and then later on when you really need miles on the board to get it to commercialisation.
Yeah, thanks, Pale. Look, I think the funding environment in Australia is changing. It has changed for the better in recent years, but it's still a big challenge. And we've got to understand that building projects like this takes a long time. We're building the cornea project on knowledge that was generated in many other projects that had to be funded. We brought together the cornea project, which still requires substantial funding to get where we want to get to. So there will be ongoing challenges in attracting that funding. But I think the collaborations also give confidence to the funders. Right? I mean, because they're funding a multidisciplined team, they're funding an integrated team where they have a high level of confidence that we've got a well-defined problem, which Jared brought. We've got potential solutions, which we're all working on, and we've got the means to deploy the solution, which Danielle just explained. And so I think funding's hard, but you have to have that integrated team just to have a chance of getting into that competitive cycle and getting that funding. And the other thing is to paint that story, which Danielle has explained for us, is that we're developing a system and a process to manufacture the bioengineered cornea. We believe that will be a platform technology for many other bioengineered products. So this investment is worthwhile. We've got the team, we've got the challenge, we've got the opportunity, and we're here to deliver on it.
Wonderful. You make it sound so easy. Danielle, question for you. We've got, as Gordon said, we've got the science, we've got the clinical challenge, but then we have a system, and the system is not yet prepared for this innovative technology because, there's a whole part of being ahead of the curve and getting this, being the first to the market. Being a 3D printing researcher myself, we went through a lot of system-wide recognition of the technology and regulatory changes that came to play to enable scale and adoption of that technology in the clinical environment. I was just wondering if you can comment on, what sort of system-wide challenges, not just, I suppose, in not only, but also in policy, regulation, but also institutions that you've required to enable this to get to that, phase where you can even dream of it coming to the bedside.
Thanks, Payal. Where do I start with that one? I think I might just start simply with, the setup that we have in terms of the tissue bank. And, it's a pretty small, humble, part of the health system, but it's at the pointy end, and we have an excellent team of medical scientists who work in that space. And really, I think the ongoing theme here is that there's always a change. We all know that there's always change, but in terms of the production of products to meet clinical need, the team are very experienced in change and adaptation, and there's something, as developments take place over time, they are adapting. We have a production team that is adapting what they're manufacturing for clinician use. We have a quality team that's working hand in hand with that production team that's challenging, and it's not always easy. How are we going to verify that product? How are we going to ensure that every single time that step is taken, that we're going to have exactly the same outcome? And when you come to automation, that's the same challenge that you need to be able to replicate over and over again. I think just moving on from that front production aspect, there is ongoing work at the Commonwealth level in Australia at the moment around the prostheses list, where medical devices and biologicals are listed and priced, and also the work around the health technology components, assessment components of those products. So obviously Australians and the Australian government wants to make sure that Australians have access to the best possible health care and the products, that are part of that healthcare, but that those products need to be efficacious and evidence-based and at a good price. So moving forward, we've been involved in the health technology assessment process for biologicals in the past, and with a product like the bioengineered cornea, which is really straddling both the biologicals and the medical device. I wouldn't say it's a straight medical device, but we're quite grateful that we have a good relationship both with the Commonwealth and the Therapeutic Goods Administration, where we can be in conversations and part of a consultative process to identify what the best way is going to be to shepherd these new products through the system. So, you know, it's not straightforward. There are complexities and there are certainly hurdles that we need to go through, but there is a pathway forward that we're able to navigate. And I see, you know, they're going to be the challenges, not insurmountable. And again, I would echo what Gordon said, you know, together we really have the capacity to deliver this.
Lovely. So lots of blood and sweat and laughs rather than tears.
A little bit, sometimes tears. Sometimes tears.
If I could just add to that, Bill, I think what we've all learned is that you know, understanding the holistic approach to the project as early as possible. So thinking for scientists to be thinking about those sorts of questions that Danielle is asking, it's really important to think about those questions early in framing the project, designing the approach, and even, you know, monitoring the progress as we go along.
Thank you so much. You know, sort of reflecting on, as a surgeon, what we've just been through, you know, the health ecosystem at the moment, which is particularly in the surgical sphere, you know, we're struggling to get cataract surgery done. And the whole system is, you know, changing quite rapidly to adopt a value-based proposition. As a cochlear implant surgeon, you know, Sydney is the global headquarters of cochlear implantation, cochlear implant manufacturing, but we know that in Australia only 10% of population actually have access to that, let alone globally. And that's such a success story. So I suppose putting that value-based perspective in your project, looking at the global need, what are the strategies that you guys, I might start with Gerard because it's, you know, I know that you see this in the hospital system at the moment. How are you ensuring, what are the strategies you're putting in place now to make sure that this technology has the maximum access across the world?
Yeah, I think that's a really important question. And I did allude to the fact that, you know, we need to basically, at the very, very least, we need to be able to produce something that's going to help in Australia. And we're fortunate because we have an banking system which is actually quite successful. But in order to get that product internationally and to actually get it to the people we need to get it, we need to have a commercial model which is going to be competitive because the market is already there. There are eye banks in the United States that are, you know, selling corneal tissue to various places around the world, and there are problems and issues and complexities and ethical issues associated with that. But that's already out there. So we need to be able to produce something that's going to be able to compete with that market. But in addition, and the reason that we actually set this program up was not to create a company that's going to make money, it's going to have an impact on corneal blindness. And so we need to have a cross-subsidy strategy which is going to allow us to get that tissue to, you know, places that, as I said, I've worked in Myanmar and Cambodia where it can do the best. But the reality is that medicine costs money. And the example that you gave first of all about cataract surgery, I mean, cataract surgery is one of the greatest operations in the world because it is very, very successful. And it is relatively inexpensive, and it has impact beyond just sight. It reduces the incidence of depression. It increases mobility. It keeps people out of nursing homes. So you can make a great economic argument for cataract surgery, which I have done many times, that it should be universally available, and no one should have to wait for it. And I've made that at very high levels, chairing committees for the federal government. But you're right. I mean, there are challenges because we have a certain amount of health funds to distribute, and someone has to make a decision about that. But you also mentioned Cochlear, which you're involved in. Cochlear is our greatest example. We see Bienco as being the next Cochlear, and we would hope that we would be able to do the same. And, you know, that issue about health care fund allocation and resource allocation is a challenge for every country in the world. But we have a strategy which is to compete and to produce something that's going to compete in the price market, but also to have a cross-subsidy strategy where we'll be able to produce tissue which will be able to help for people who might not be able to afford the ones that we're sending to, and places that can afford them.
I'll just add, Pale, just in terms of that accessibility, I think another important part and important challenge for the scientists, engineers involved in this project is to make sure that whatever we're designing in terms of manufacturing, of course, is innovative, gives us a high quality product, but also that it's deployable. It's deployable manufacturing, because I think that will be another important aspect of the accessibility of this technology around the world.
And so, in terms of the design of the printers and the devices, it's going to have an on-off button. Is that what you're saying, Gordon? It's going to be easy to use at point of care.
It'd probably be run by scientists and engineers rather than clinicians, so we probably could get
more
away
than
with
on
a
and
couple of
off.
buttons.
That's outrageous.
And if I
Yeah,
might,
I was actually going to come to you in terms of so please go, and
So
I have a question for you.
If I might just go back again a bit, back to the beginning of all of this, and we talk about that those countries globally that where people aren't able to access treatment for corneal blindness, and the fact that tissue is scarce and treatments rely on tissue banks and deceased donation, and we just really step way back from automation where we are now, and the fact that for each corneal transplant we have someone calling a family who's just lost a loved one, asking them whether they would be prepared to donate their corneas. That if that family, and probably 5 out of 10 families might say yes to donation. So if that family says yes, and that those corneas are assessed as medically suitable, then that at best we have 2 people treated. With the work that the team has been doing, we think that we're going to be able to produce 30 bioengineered corneas from one donor cornea, and that's a really great step forward. Then when you layer on top Gordon's automation, we're really in a place where we're able to provide or ensure access to treatment for many more people.
I'll add one more comment if I may.
Yeah, of course.
So the other cost is always an issue in terms of production, as Danielle and Gordon have articulated, but the other advantage, and this is one of the advantages that we found out along the line, along the journey, was that the cost of getting human collagen to build the cornea, which is obviously the major part of the cornea, is incredibly expensive. And so rather than having to purchase that or get it elsewhere, because we are part of the New South Wales Tissue Bank, we're able to actually get human skin and placenta, and human skin, which is obviously opaque, and turn that into high quality, transparent human collagen for our use in building the cornea. And so that is, I think, a wonderful way of not just celebrating the gift that people have donated, but it's also is a way that we can actually sort of make that product that we're going to be getting to market much, much less expensive.
That's wonderful. I have so many questions, but there's one from the participants. Fantastic initiative and a great team. Just interested to know whether there are other competitors working towards producing a similar situation, and if so, can the project be accelerated to bringing it to market earlier? And I think you did cover that somewhat, didn't you, Danielle?
Yes, I did. So just by way of summary, there is a lot of interest in this space, and we certainly feel some pressure to be first to market. However, our competitors are predominantly just focused on one layer or the xenografts, and we are the only group that's working on the multi-layer, full thickness bioengineered cornea. So we have some confidence in that regard, but not complacency. We're working hard. We really recognise the importance of being first to market.
Exactly right. And 2 other comments I'd make. And one is that we are competing in the space of the partial thickness transplants as well. And we would expect to be well within animal trials within 12 months for that product. And that's obviously an important part of us building the total cornea. But, if there was no competitors in the marketplace, as someone once told me, then it probably means there's no market. And so the fact that there's competitors doesn't scare us. It just means that A, there's investment available, and B, there's a need for these products. And so we relish the competition.
Yeah, and also that means there's awareness. There's more people, more companies building awareness amongst policymakers globally to have the policy aligned. I suppose the other aspect of the question is the demand. One of the difficulties about is also, patient awareness, consumer awareness. What you don't know, you don't know. And so, how are you addressing the issue of bringing the patients along the journey in your project?
This, as well as the knowledge that each of the groups bring, this has been a great, learning curve for all of us. And we've been participating to different extents in commercialisation, education, and incubators and accelerator programs. And what we're doing with both with consumers and with surgeons is that we are talking with them and talking broadly and extensively to try and understand what surgeons want, what patients want, to talk about what we think that we've got, and to hear from them about what they think would be the strengths, what else they would like. Perhaps there's something that we haven't thought about. So we're really seeking extensive feedback from both patients and surgeons and others, really. I mean, we will also at some stage be talking, to those, the payers, the health insurance companies, and to government along that way, because it, as I referred to when I was talking about stakeholders, we are really cognizant of the fact that we're onto something good here, but we don't know everything, and we really do need particularly to talk to patients and to surgeons about what they think would contribute to an outstanding bioengineered cornea.
And if I just add to that too, Pell, I think everyone in the team, everyone from the youngest researchers through to the oldest researchers, really appreciates that aspect of what we're doing and is committed to putting the effort and the time into being engaged in those conversations. And I think that's a great part of being a researcher in an interdisciplinary team like this, is that we learn to communicate with those people who are going to be the users and learn to listen to those and put that energy into that type of activity as well as our research early on in the project.
Wonderful. We've got some more time, so I'm just going to throw a question to Gerard, just because I see this in the clinical sphere. Normally we have, for most procedures, we have clinical guidelines which tell surgeons, when to operate. When you have something new coming on that's so unique and so innovative, where is the ophthalmology world in being aware of this, or even developing, what are the guidelines currently about corneal transplantation, and how does this fit in?
Yeah, look, I mean, corneal transplantation, as I said, the first one in Australia was done in 1940, and I think it's fair to say that things have improved significantly since then. But in terms of the guidelines and the regulations around the actual corneal tissue delivery, Danielle would be the expert there. But from a surgical point of view, yes, of course, we have guidelines, we have indications, we have peer-reviewed, we have clinical care standards. Our college, the Royal Australian New Zealand College of Ophthalmology, sets out guidelines for all of these surgical procedures, as I'm sure they do with the College of Surgeons. And I wrote the ones, or was involved in writing the ones for corneal disease and surgery. So that's already established. I think that the challenge for us is going to be able to prove that what we're producing is at least as good as what's currently available. And that will be the challenge. And of course, we will need to go through the usual process. We've already, as I said, established some protocols for some animal trials. We need to go through all the clinical phases until we get to our phase 3 trials before we can even think about using this or distributing it for general usage. So it's a relatively long road, but it's an appropriate road because we can't have anything coming out and being used unless we go through that process. There'll be further challenges in terms of the European CE mark usage, as Danielle alluded to, and then of course the challenges of the FDA and the United States. But, you know, one of the first products that will be coming out, so that apart from the full thickness cornea, as I said, there are a couple of other related products that we will be producing, and the first one will probably now be that inner layer, just transplanting the inner layer. It's called an endokeratoplasty. And there's been many publications now that have shown that if you can actually do that, that will actually be less expensive than what's currently available on the international market. And so apart from it being better clinically, apart from it going through the appropriate process of clinical trials to prove that it's effective, we also think that we can do it in a way that's less expensive than some of the other products around. And as we talked about earlier, I mean, cost is a factor. Whether you like it or not, money and medicine, they're related, and even if they make uncomfortable bedfellows, it's part of the equation.
And so when do you think that this is going to be coming into clinical trial phase?
Well, we've already sort of got a plan which we have submitted to numerous funding bodies, and I would like to point out that we were successful in the round 1 of the MRFF, and that's put us into the round 2 of the MRFF, and we are hopeful that we will get some significant funding to take us forward. But regardless, we will be moving ahead. But I think that it's within 3 years we're going to be in clinical trials for our endothelial keratoplasty. The full thickness transplant will take a little bit longer, as Danielle alluded to.
And so, Danielle, how are you preparing the whole process for that amazing first but very difficult step, a clinical trial?
Well, I think again this will be a collaboration, and with Gerard through the medical schools at the University of Sydney, through the local health district where the organ and tissue donation service is hosted, South East Sydney Local Health District, will be working together on those clinical trials, and also with our other partner, the Centre for Eye Research Australia, has some expertise in clinical trials. So I think I can't imagine there's anyone listening to this webinar who wouldn't be well aware that you need to lay the foundations for this sort of work many months, if not years in advance. So those conversations, again, with partners are underway. Is this something, well, where exactly will we be doing this? And I think we will be determining the location and how we're going to roll out the clinical trials as we learn more, as we refine these final processes of the protocol of the bioengineered cornea, and also the EK we're referring to, the endothelial keratoplasty product. And there are a number of different products that we expect that will be generated along the way to the full thickness cornea.
I love your strategy, how you've sort of gone simple and then built up the complex, and each project then builds on the other. So we're sort of almost at the end of the time of the webinar, but I'd love to get some final comments from each of you. Any one-line words of wisdom to walk away with?
It's really hard.
More tears than laughs.
But it's worth it.
Lovely.
Yeah, look, it's challenging, but the opportunity is so huge that it's worth taking on the challenge, as Gerard alluded to. And just add to the comments around the competition that Danielle was talking about before, and our competitive edge. I think the real competitive edge we have, besides all of the other things that Danielle explained, is in the strength of the collaboration and the commitment to those final products. And that's not just at our level. That's through the whole team in terms of commitment to delivering on this challenge and delivering together.
Then that's the key, isn't it? It's just the people that make it rather than the idea necessarily. Danielle?
That's right, and I think, look, I might just go off on a little tangent as part of my final words, but following on from Gordon's reference to people, next week is Donate Life Week. Even the bioengineered cornea wouldn't be possible without people agreeing to donate their organ and tissue for transplant. In Donate Life Week, we encourage all Australians to consider their decision around organ and tissue donation and to register their decision. They can do that on the Medicare app or even do it in less than 1 minute at donatelife.gov.au.
Thank you, Danielle. Look, thank you so much to all of you and also to all of our participants for dialing in this afternoon. It's just been a really fantastic afternoon. I feel very inspired and I'm going to keep going and working harder on my own research. Thank you all.
Thank you.
Thank you.
Thank you.